would like to start from thanking the organizers for an opportunity to share with you our progress on this exciting project, which is focused on transcriptional control of Golgi biogenesis. And Golgi is really a fascinating organelle, which is composed of several compartments. This Golgi compartment is involved in reception of a secretory material which arrives from the endoplasmic reticulum to the Golgi. Then medial compartment or core Golgi region is organized in stacked cisternae, and they work as a manufacturer for processing of proteins and lipids, which then leave the Golgi to their final destination from trans-Golgi compartment, sorting the proteins to the right places in the post-Golgi space. So during their life, cell in many situations need expansion or generation of a new Golgi. This happens, for example, during cell cycle. Then the mother cell has to provide the daughter cell with more Golgi membranes. Then the cell has to handle increased secretory load, moving across the secretory pathway. And during differentiation, then such cells as neurons or muscles generate new Golgi units. The problem is that transcriptional regulation of Golgi biogenesis remains poorly understood for several reasons. First of all, this issue was kind of neglected by Golgi field for decades, and the field was focused on entirely different things. Then it's difficult to study the transcriptional responses during physiological situations described before, because these responses are contaminated by expression of mitotic genes or cell-specific genes, or genes involved in ER stress or unfolded protein response. So ideally, one would like to have the system for transcriptomic study, then Golgi forms from scratch, and at different stage analyze how the transcriptome is remodeled. Unfortunately, such system was missing, and we tried to invent it. To this end, we generated HeLa cell line, which express Golgi enzyme mannosidase 2 linked to horseradish peroxidase. Here you can see Golgi highlighted in green in these cells, and then we add to the cells H2O2 and diaminobenzidine. Diaminobenzidine is deposited in the lumen of the Golgi, and in this case, it kills the Golgi with nano-surgical precision. So our bet was then that Golgi will recover, okay, after this harsh procedure, and this was actually the case. Here we can see the Golgi was inactivated in a couple of cells. You can see this diaminobenzidine DAG deposit, and the cell managed to make the new Golgi appear as a green over the time. It takes pretty long to build the new one, because 6 hours after inactivation of the old Golgi, we found new Golgi units, but we call them kind of ugly Golgi or imperfect, and only by 10 hours the cells managed to recover the bona fide Golgi stacks. So to summarize characterization of this system, which took us a couple of years, I made this timeline. At 2 hours, we start to observe first detectable Golgi structures. At 6 hours after inactivation of the old Golgi, we have what we call ugly Golgi or Golgi precursors, which, however, are not perfect morphologically and also fail to support trafficking of the protein across the secretory pathway. And only by 10 hours, we observe formation of polarized Golgi stacks with perfect architecture and capable of supporting Golgi trafficking to different destinations. At 18 hours, we saw more cells making a perfect Golgi, and we wanted to understand how the transcriptome changes during this process. To this end, we collected the cells from each time point and then subjected them to single-cell RNA-seq. Why single cell? Because we observed a very heterogeneous recovery phenotype across the cell population. Here you can see some examples. This shows perfect recovery of the Golgi because we saw both key markers recovering in the Golgi area, green and red. We also observed partial recovery, only green marker, no recovery, cells without newly forming Golgi. We also observed dead cells, and the cells where mannosidase 2 was expressed at the lower levels, so they were not inactivated and were doing nothing. We obtained these UMAPs with clusters of the cells and started to analyze each cluster for gene enrichment with gene enrichment ontology analysis to understand whether any contain enrichment in Golgi genes. And this was the case because cluster 2 and 10 has enrichment in Golgi transcripts, and this shows the example of Golgi genes over the UMAP with high expression in the cells corresponding to cluster 2 and 10. Now, we also confirmed transactivation of Golgi genes during this process using fish. Immediately after transactivation, their transcripts were low in number. You can see them in red. However, 18 hours later, we see the recovery of the new Golgi in green, and this was paralleled by appearance of large number of transcripts. So we had Golgi activation of Golgi genes. So questions which we asked then were quite important for entire project. First of all, we wanted to understand whether among transactivated Golgi genes we have some prevailing group. For example, structural genes or trafficking genes or any compartment, cis or trans. And we also wanted to understand whether they are activated in some order. And the second important question was how this activation correlates in time with structural and functional maturation of newly forming Golgi organelles. And first of all, in terms of compartment, we found some prevalence of trans Golgi genes, but that was not significant because also cis and medial Golgi genes were well represented. And the same can be said about also different functional groups. The most important and interesting finding which we made was related to kinetics of Golgi gene expression, because we found that most of Golgi genes which were activated switch on between 6 and 10 hours. And now, if you remember, 10 hours is exactly the time then we had the recovery of Golgi structure and function. This means that contribution of Golgi gene activation is needed for structural and functional integrity of newly forming organelle. Now, we also found that this was the same trend in terms of kinetics for different groups. So independently from cis, medial, and trans, they showed this switch on between 6 and 10 hours. And at this point, we can say that the massive transactivation of Golgi genes, first of all, correlates with recovery of structural and functional abilities of Golgi organelles when we started to form from a scratch. And also induction happens in a very coordinated manner, independently from function or compartmentalization of gene-protein product. This coordinated activation suggests that there is a master regulator, transcription factor. And to find such transcription factor, we implemented bioinformatic workflow, which as an input used a set of more than 100 upregulated genes in our single cell set. So we identified common transcription factor binding sites in their promoters, and on this basis predicted the transcription factors. For the candidates, we studied the expression in a single cell dataset during biogenesis of a new Golgi organelle, and discarded the candidates with poor expression or whose expression was reduced over time of Golgi recovery. This left us with candidates highlighted in pink, and we implemented a RNA-based screen to see whether their suppression would impact recovery of the Golgi organelle. To do that, we silenced them and then inactivated Golgi and start to see where the new Golgi recovers. Here are examples. These are control cells before inactivation with green Golgi. We kill the Golgi. It starts to be black and dark. And after 10 hours, control cell managed to recover Golgi marker quite efficiently. This was not the case for example, suppression of Krebs3L1. And we also found other transcription factor with the same trend. Then we validated them by electron microscopy. And this was pretty striking that in control cells, we had a recovery of the Golgi stacks, while, for example, silencing of Krebs3L1 severely impaired recovery of the Golgi architecture, indicating that it's maybe involved in activation. The same was found for MITF and SP1, and for sake of time, I don't show that. Finally, we decided to analyze whether silencing of this transcription factor also reduce or inhibit transactivation of Golgi genes. And we found that indeed this was the case for Krebs3L1, while suppression of MITF or SP1 had a moderate or no impact on Golgi gene activation, indicating that Krebs3L1 works in Golgi biogenesis through activation of Golgi genes, while other transcription factors regulated through different, maybe indirect mechanisms. That was further investigated in terms of Krebs3L1 involvement by searching the Krebs3L1 binding sites in the promoters of our activated Golgi gene set. And we found many binding sites in our set of Golgi genes. Moreover, we also observed the nuclear translocation indicating activation of Krebs3L1 during the process of Golgi biogenesis. Immediately after inactivation, it was mainly cytoplasmic, and then it's moved into the nucleus, and the peak of a nuclear translocation was observed at the 6 hours, and this is exactly the time which precedes transactivation of Golgi genes, which makes perfect sense. So this suggests that Krebs3L1 could be a master regulator of Golgi genes. And to test this, we thought just simply overexpress it and see whether we will have an increase in Golgi transcripts in HeLa cells. So here you can see volcano plots for overexpressing cells, Krebs3L1, its constantly active form, MITF and SP1. Then we analyzed let's say, activated transcripts for gene ontology enrichment terms, and we found many Golgi annotation terms in the case of Krebs3L1 overexpression and its active form, but not for MITF or SP1, indicating that indeed Krebs3L1 could work as a let's say, master activator of Golgi genes, And that's the program of Golgi Biogenesis is likely to be driven by this transcription factor. This opens a new avenues and questions to understand. For example, can this transcription factor be used for tissue regeneration or engineering, whether it may be hijacked by pathogens to invade the cell or for their replication, whether it could be helpful in the context of medical biotechnology. For example, by our expression of CREP3L1, we could force the cell to generate more enzymes which can be used for replacement therapy. And finally, whether this program is somehow modulated during diseases related to Golgi complex dysfunction, and whether it can be targeted in this context for development of therapy. With this, I would like to wrap up my talk. I would like to acknowledge the members of my lab and collaborators within this exciting project, Jan de Boer and Alexei Khudyakov. I would like also to thank Frank Pirez, who provided us with a number of advices and reagents. Two people who contributed most, Francesco Formo and Domenico Abete. And I would like to make a specific thanks to Human Frontier Science Program because they believed in this project, and without them I couldn't realize it for sure. Okay, thank you.